Okay, welcome back to our lecture series on single-cell high-throughput data science. Today is the topic number 10. We are going to talk about the construction of single-cell gene regulatory networks. So people have been using functional network analysis for different domains of the study. And here is an overview of the data structure, and you will encounter it during the functional network analysis. This particular example is from a fMRI study, and we can collect the information from signal from different regions of the brain. For example, one is in orange, and then another is in this blue signal. And you can collect signals over the time, so you have a time series signals sampled from different regions of the brain. If you have enough of those samples and from a different area of the brain, and you can construct a correlation matrix if the signal from two areas they are highly correlated to each other. For example, like this orange curve and blue curve, there are certain levels of the correlation, and they are, although the physical distance between them may be not so close, but you may recover a correlation among the signals they produced. So in this way, you'll create a correlation matrix, and then you can apply some kind of cutoff to get rid of the weakly correlated correlation and pairs, and then you end up with a so-called adjacency matrix. So this is a network. Okay, adjacency matrix is a matrix representation of network. So once you get the adjacency matrix, you can reconstruct this network, and each point here is one of the region you just sampled, and the link between them indicate their correlation between two regions. Okay, so this is an overview about the process we can use to generate functional networks. When you have the single-cell RNA-seq data, then you have an expression matrix for many different genes among many different cells. Here you have multiple samples, which is a cell. Okay, so from cell, you can construct the correlation between genes. So that when you compare the gene from one to another, you can come up with this correlation matrix. And then the downstream analysis will be the same. And then you just apply this procedure and to generate a functional network. Now, the node in this functional network will be the gene. And then the link between the gene indicates the two genes are co-expressed, for example. So that's the overview about the functional analysis using single-cell RNA-seq information. Okay, nowadays we have information from single-cell multi-omics. You can have gene expression, you can have epigenetics information from single cells. So that's gave you opportunities to construct different types of networks. Different flavors of this. So in this review paper recently published, the authors lay out a couple of terminologies that have a different type of the network. So gene co-expression network is one of the most commonly used network, because you can study the correlation between gene expression across samples. This sample could be single cells, okay? Then you have a gene regulatory network. This is a construct between transcription factors and the downstream target gene. Okay. If we didn't mention this single cell attack-seq in our lecture series, but this is a so-called chromatin accessibility. So attack-seq gave you the information about the chromatin accessibility. So if you have a transcription factor and the binding site, for example, and then you have an open accessibility, access to open chromatin structure, then you can study which pairs of gene at the same cell share the open chromatin structure. That indicate these two genes are co-regulated. So that's the information, and you can use that to construct the so-called cis co-accessibility network. That's something new and proposed by those authors. Similarly, if you have a methylation information, you can do the methylation signal association gene regulatory network. And then if you have a so-called high-C analysis, and nowadays you can do single-cell high-C analysis, and then you get the spatial, three-dimensional spatial interaction information that the chromatin interaction network can construct. And then there are cis regulatory element, and the gene interaction network can be constructed if you combine the information from single-cell RNA-seq and the single-cell attack-seq. Finally, you can have a transcription factor against cis regulatory network, cis regulatory element interaction network. And also you can somehow combine all this network information. If you collect this information from the same cell lines, same sample, you can combine them. If you use single-cell RNA-seq, single-cell attack-seq, and the methylation sequence, and the single-cell high-C, and you apply all this technology to a single sample of your cells. Then you can combine the different signals to build those different networks. For example, gene co-expression network can construct using information from a single-cell RNA-seq alone. And you can have this gene regulatory network if you know the transcription factors and their targets. And if you have a attack-seq, that epigenetics information, you can have this co-accessibility networks. You can construct network for methylation and transcription factor binding, and use that information because you have those open access information, and then once you have a high-C, you can construct a different type of the network. Okay, let's go back to some basic terminology regarding to the network analysis. So you can call it graph theory, and there are also you can call it a network. So in here you have a node or links if you think this is a network. In graph theory, we call it vertex and edges. Okay, so the most important matrix representation is, as I mentioned, adjacency matrix representation for a network. For example, if you have a network like this, your adjacency matrix will be this. Okay, you have 5 nodes in this network, and then adjacent matrix will be a 5 by 5 matrix. Each row or each column is a node. And then if node 1 and 2 are linked, then you have this position as 1. And this is undirected network, so both 1, 2 at the 2, 1 position of this matrix is a non-zero value. And for example, 2 and 4 are not linked. So the position of 2 and 4 should be 0. So 2, 0, 2, and 4, or 4 or 2 are 0, because that indicates these 2 nodes are not linked. So that's very easy to understand, and that's the representation of the adjacency matrix for the network. And then for the network theory, and we need to mention the centrality measurements. So once you have a network given, then the first question you may want to ask is which node are the most important node in the network. So in order to measure that, you have to use a set of so-called centrality measures to give you quantitative measurements of the importance of this node in a network. So over the years, people have already invent or create a lot of different types of centrality. You see in this example, we have a closeness centrality, we have a class ring coefficient, we have a degree. Degree is the simplest centrality we can use to just indicate how many partners of this node are linked with. So if this purple node have 4 linkers with other nodes, the degree of this is 4. And we have a betweenness centrality. These all are very commonly used centrality measurements for the important to quantify the importance of nodes. In these small examples, the node give you the color and their centrality indicators are the node have the highest centrality of that type. For example, the purple one have the highest degree centrality, and then this blue have the highest betweenness centrality among all the other nodes. And then this red one have a highest closeness centrality. And I will quickly mention how this, for example, some of the centrality measurements are calculated. I use the closeness as a first example, and this is the equation to compute the closeness centrality for x one of the nodes. And then the definition of this centrality is the sum of all the distance between the node we are measuring here is x, the distance between the x against to all the other node y all over the network, and the sum of this distance divided by 1, and that's the computation of closeness centrality. And here is a very simple example. We are going to compute the closeness centrality for A, the node A. And then you compute the distance from A to B, that's one step, so there's only one. So distance from A to C, the 2. A to D is 3, by 3 steps, 1, 2, 3. A to E is 4. So that's the A under consideration, and its distance to all the other nodes. And then you sum the distance, and then use 1 to divide it. And then that's the closeness centrality for A is 0.1. Similarly, you can compute the closeness centrality for B, and then B have a centrality 0.14, and C have 0.16, 0.14 for D, and 0.1 for E. And if a node like C is in the middle of the network, then C has a highest closeness centrality. So that indicates C probably is the most important ones in this small network, which is only a linear link between all the nodes one by one. Similar idea, we can compute so-called harmonic centrality. Here we replace the distance, average distance, or sum of distance with a harmonic means of all distance. And that's the equation. And in a similar way, you can just compute harmonic centrality for all the nodes in a network. And here, this node has the highest harmonic centrality among this sub-network, fully connected sub-network. Okay, so the next matrix we are going to, the next index for centrality is so-called the COTS index. It's a type of spectral measure, and then measure the influence by taking into account the total number of the works between pairs of nodes. So to compute COTS index, we need to take the power for the adjacency matrix of the network. So this is adjacency matrix of the network, and then you take the power to 2 to 3 up to infinity in order to compute the COTS index. When the power, the k is large enough, and then your solution will converge, and you can compute the sum of this. So that indicates the total number of the work of the length k between x and y. So that's k is here, and then you summarize all of them to give you the total number of works between from one node to another, and you calculate the influence rather than use the distance between two nodes. You use this total number of possible works. And so in summary, you can use different types of the centrality measures to evaluate the so-called importance of each node. And for example, in this network, you can use degree centrality or closeness centrality, harmonic centrality, or COTS index to measure the importance of the nodes, and you may get different answers. So for the same network, for the same network, in if you use degree, you found a cluster of the nodes looks more important in this group. However, if you use closeness centrality in this panel B, and you see the highest value of a closeness centrality is among those nodes in the center of the graph. If you use harmonic centrality or use causal centrality index, you get different measurements. Centrality measures are indexes to be used to measure the importance of nodes. Next, we are going to explain the co-expression network of the gene. So the most important package, the method people use is so-called WGCNA. So that's the name of this package. This package has been widely used in the gene co-expression network analysis. It's R package and have different steps. The first step is will construct a gene co-expression network. And then after that, you will use this constructed network to identify the modules. Modules are a set of genes which in this co-expression network, they are close to each other and form a co-expressed module, and then they are perform or behave similarly regarding to their gene expression, and then you can group them. After that, you may want to use external information, and you have a clinical data or SNP or other information to link or with the module you detected and see, for example, particular module contains gene. If they have a high expression, then you have a certain clinical phenotypes. So you can use this information to do the downstream network-based analysis to link with your biological signals. And you can also study downstream and the module relationship and also other maybe comparative analysis. So the most important steps of this package is to construct gene co-expression network. So that's step one, is the most important step. And then you have module, and then you do module-based exploration of the correlation. So let's see how this first step is conducted. So imagine you have only two samples. One is control and another is case. Then you will find some genes expression upregulated. So those three orange genes get upregulated in the case compared to control. Well, this blue gene has no change. And then green gene, three green genes downregulated. So that's very highly simplified case. And if you have multiple samples, you may do the regression, and instead of just high or low upregulation or downregulation. But anyway, you can find a set of genes which, like the set of orange or set of green, they behave similarly and in the consistent way across samples. So just rely on this relationship. You can construct a correlation matrix between genes, like three orange genes behave similarly, so they have a correlation to each other high, and those green genes have a high correlation to each other, but not to other genes, and those blue genes. So rely on this correlation matrix, you can translate this information into a co-expression network. And among this, you see those genes in the same clusters and then behave similarly. So that's the group, that's another group, and that's probably 3 groups. So that's the principle behind this co-expression network. In reality, this in the real biological signal is highly noise, and you have a lot of false positive, false negative probably. And then there's a way, and WGCNA adopted so-called Tom similarity matrix, and to use the concept of topological overlap matrix to denoise, basically. And that's the original adjacent matrix from the correlation. And then after you use this topology overlap matrix to replace your original untreated raw correlation matrix, you get most smooth signals, and then you discover these clusters of genes which cannot easily identified from the original correlation matrix. And they are the idea was borrowed from a science paper published by in Science by Bob Barcia, and they created the concept which called topological overlap. And the author of the WGCNA borrowed that and use that as a smoothing approach. So you can specify the level of the smoothing, and then you can have a slightly different, you change this level of smoothing, and you have a different discovery of the network modules. So in this case, you have this module, and then you smooth this out, then you have a larger module. At the end, you will choose, say, what the best topological overlap measure that you want to use, and then you get the cluster of the genes. And then that's the output for WGCNA. You see these clusters of the genes, and you can merge some of these clusters and make them more make larger gene sets, and each bar black line here is a gene, and this is a hierarchy clustering tree, and each color represent one cluster, and then the bottom panel is merged clusters. All right, that's WGCNA. As I said, is the most commonly used package in RNA-seq analysis, including mostly in the bulk RNA-seq if you have only a couple of samples. In the new era of single cell, and we have information from multiple cells. So this gave our power to construct cell type specific, and also because of the sheer number of cells we can sequence, and this allow us to create a highly reliable network of gene expression. Well, there are many advantages using single cell data to replace traditional gene expression data to construct the network. See, so-called bulk expression data return average expression of gene among large number of cells. So you lost the function, the cell type as a function of your expression. So you don't have that information and use bulk RNA-seq data, for example. In the single cell, on the other hand, it provide you the cell type specific information. Another advantage from single cell data is you have a large amount of numbers of cells. So that gave you the power to establish a relationship across genes, which you just cannot get so many samples in traditional bulk RNA-seq. And in bulk RNA-seq, you rely on multiple replicates or maybe time series samples, but you just cannot get enough data points in most of the cases. And also, they'll measure the gene expression directly from individual cells, give you all the necessary information to directly infer gene regulatory network. And that's so that's difference has been illustrated in this figure, and then that's a single cell. And if you use traditional bulk data, you pull the profile and those genes expression is a reflection of the global average. So you don't have a cell type specific information for this gene regulatory network. In a single cell study, you can classify and identify cell types, and you build the network specifically for different cell types. So you see the different colors indicate the gene 1 and 3, the regulation between them, the correlation between them is only happened in this population, but not in other. So that's the power, and this summary of the advantages. Single cell data sets are large, thousands of cells, and the single cell data inherently provide variability required to detect significant statistic relationship between genes. If you have a traditional samples, you may have a population-based study, and that need introduction this variability using perturbation, or you have to rely on multiple samples at different time points. In single cell data, as we showed in pseudo-time analysis, even you sample your cells at one time point, it still contains the variability because there are synchronized cells in different states, and you can rely on this nature biological heterogeneity, and then to use pseudo time analysis, for example, to construct this to give the information of this different stage of to give you the variability. If you have enough resource, you can certainly collect the single cell samples from the different time points. That will give you the time series data, and also for single cell. So this figure illustrates the use of the information from a single cell to construct the network, and we some analysis pathways you can take. You have input single cell profile, that's matrix of gene expression. You can do the clustering analysis to identify the different clusters of cells, and then you build cluster networks for different networks for different clusters, and you can compare as well. You can do trajectory analysis, Pseudotime, to range cells by their Pseudotime values, okay? And then you can build a dynamic network using some inference. And you can certainly, I forgot to mention this, you can treat the whole population as a whole, and then you construct a static network. So just a fixed network. There's no different part of this. There are profile-specific network, and we will mention the scenic. Okay, the profile is from the known transcription factor and the target relationship to help you to get your network which specifically reflect what we already know. There are many different methods have been developed to do this. This table is just some examples of the tools and the algorithm you can use for single cell network inference. This illustrates to construct or infer dynamic network. You have single cell data, and then you identify the structure among cells, and then put a trajectory on the cells and older cell along the trajectory to use it as the pseudotime index. And then from there, you have this information that's built from the single cell itself. Then you can use this to get a temporal aided network inference. You can build a dynamic network along the trajectory. There has been so many different methods. So there's a specific study which they compare this different algorithm and to do a benchmarking analysis, see which algorithm is good in which scenario. So the whole evaluation pipeline is called a B-line, has been published, and then include the simulated data and the real data as a test. And in the uniform package framework, you can run different algorithms and then generate the predicted network, and then do compare to the known network structure and evaluate using different metrics. So this is a summary of different algorithms, the single cell network construction algorithms, and their performance reported by B-line study. The network they use, for example, this the network they used relatively small because you have to allow so many different algorithm to run on it in a limited number of this gene can be involved. So although this is a systemic evaluation study, but the shortcoming of this, the limitation of this study is the testing network just too small, content too few genes. Anyway, so this is a study they used, and this the results, summary result, they had the accuracy and the specificity. They use different metrics to evaluate those methods. So we will spend some time to go over several of those methods. One is the gene3, another is the tSNE, which is based on the gene3, and the PIDC, score, scrap. So we'll quickly go through some of these existing algorithms. Gene3 is published in 2009. It's in a competition called the Dream4 competition. This algorithm compete with a lot of different algorithms that turn out to be the best. The Gene3 is based on random forest. It started by generate so-called learning samples. Okay, so that's a lot of mathematical details. Basically, you generate different sets of the so-called learning samples, and you are going to use this to train the algorithm, the random forest algorithm. So for instance, this is a learning sample 1, and then this is output gene expression. So, and the rest of them are the input. So this subtask is to use all the other genes expression to predict this first gene's expression. The method they use is a random forest tree assembly. And then after that, you can rank the genes, see which gene provide the most substantial contribution to predict this gene's expression. So you have a ranked gene list. Then you move to learning sample number 2, and you learn to use all other genes expression to predict the second gene's expression. Use a similar method, you construct another rank, which is for the second gene. So after you finish the loop for all the genes, N genes, you have an N profile of this. And then you integrate of this gene rank profile of the prediction power for each gene. Then use this interaction ranking to construct the network. Okay, so that's so this method eventually is based on the random forest, and this process is computationally intensive. So Gene3 will take long to run. tSNE in the core is based on the Gene3, and they add this transcription factor binding motifs analysis into the results of the GeneSRI. So if you have the two genes are linked in the network by using GeneSRI, scenic will search for the regulatory relationship between these two genes. If it's identified, this is a transcription factor and this is its target, then these pairs of link will be kept and used for the downstream analysis. So scenic itself is not an independent algorithm to construct the network. It relies on GeneSRI to construct co-expression network, but their downstream analysis have been included in the scenic workflow, including to identify the regulatory network by restrict to the database support transcription factor and the target relationship superimposed onto this co-expression relationship between them. And then their downstream regular activity identification. So this workflow has been applied in the real data analysis, and then they demonstrate it's very useful to identify regulatory networks in this example mouse brain sample. The next method is called partial information decomposition. So this is based on the information theory and the package has been implemented in Julia language. It does not require cells to be ordered by pseudotime, and no extra information is required. It's based on this information theory to compute partial information decomposition method to be used here. Another tool I want to mention is called scRNA. It's R package, and then it rely on pseudotime information. And it optimizes two parameters, W and B, and in this differential equation, and then reconstruct regulatory network using this. The last package is called the script, can be used to infer the causal gene regulatory network. It applies to learn both linear and nonlinear causality, and to recover causal interactions. And next, I'm going to mention the multi-layer network analysis. So this is this kind of analysis will be really useful once you have a network can be built from a different time point of samples. You can sample cells along the pseudotime and reconstruct multiple networks, and we call this multi-layer networks. And then among the multi-layer networks, you can identify the modules So modularity, there are algorithm to compute this. For example, you have in this equation, we have a gamma and omega. Then one is for the strength of topology, and another is temporal connectivity. And to build in these equations to give you the multi-layer modularity measurements. So if you try to change the values of these parameters to either maximize or minimize this Q, and you can get different solutions of the modularity across time point, and then you can discover the network switching events. This shows you some network switching events. You have two networks. One is in the red nodes, and another is among blue nodes. This blue genes interaction get switched. There's one red gene in this module along the time and switch to the blue module. And then at time 3, you see the change of the membership of the different modules. So this creates a scenario, a situation which you have a network switching. So we would like to identify those network switching events. Then maybe those events are important to explain the differentiation of the cells or change of the cell states. Their algorithm can be applied using time varying graph theory, and then you are considering here a time varying graph, and there are different steps of time, and then you reconstruct from this multi-layer network into a profile network. So this is called aggregated static graph. The total contact duration through all the time course, and then you found the change of the weight of the links. The last approach I would like to mention is the software is called Con mode. It's use non-negative matrix factorization, and in the specific way to take into account of the change of the modules across time. To do this non-negative matrix factorization learn the features to identify those conserved modules which are unchanged across time points. Those are the advanced topics, and it will take long to teach or to learn. And that's the end of this lecture, and I appreciate your patience over the time, and hope we have another chance to study together. Thank you so much.